Hello guys, my name is Thang and this is my friend Thang, and they know our names. Today we're going to be talking about a few processes that are necessary for the human body. What do you think they are?
DNA replication and protein synthesis. So let's not waste time here and let's get straight to the video. Let's get this over with.
Alright, let's go.
Let's direct our focus to DNA replication. Now let's refresh our memory. What is a DNA structure again? Now you can think of a DNA structure as a chain of nucleotides making up of a phosphate groups, a pentose sugar, and specifically since we're dealing with DNA, it's a deoxyribose, and connecting with that will be a nitrogenous base. Now there are four important bases that forms up majority of the DNA structure and distinguish one from another. You have A for adenine, T for thymine, but if you're dealing with an RNA structure, for example, instead of thymine you have it replaced with a uracil, and C for cytosine, and of course G for guanine. And these four bases are going to be very important for the basics machinery in terms of the complementary pairing. Now, the mechanism of matching bases is very important in DNA replication. Now remember, the four bases T, G, A, C are pretty much in a complementary couple almost. You can think of this as, for example, a couple with traits that are specifically meant for one another, where one of the couple will have a nitrogenous base of A and the other partner, on the other hand, will have nitrogenous base of T, and they match up in similar way as it is within the DNA structures. Now, you can even think of this as the key point to the lesson. To even begin the process of DNA replication, you need to know the following parts, such as the topoisomerase, which is an enzyme, and another enzyme called the DNA helicase. And these two parts are the basic fundamental machinery within the process of unzipping a DNA strand. Now, first off, before anything really begin, you would first have your DNA strand as laid out. This is when the topoisomerase will comes in and begin cutting the phosphate backbones, and this will untangle the DNA, making it much easier for the splitting as well. Once the topoisomerase has gone over, the DNA helicase begin going through the DNA and cutting as well as splitting the DNA strand into two individual and smaller strands. Once this is done, you now have two individual strands, and one of them will be called the lagging strand, and let's call the bottom strand the lagging strand, and one of them will be called the leading strand. And there's a distinguished difference between the two strands. But first, let's focus our attention towards the leading strand. Now, to begin the polymerization process, first you would have a DNA primer, primase, which is an enzyme which is going to go in and install a small RNA segment, which is, you can think of this as the initial point to begin with. And this initial kind of starting point will begin the polymerization process. Once you pretty much have this primer set in, another structure which is called DNA polymerase, which is another enzyme, is going to go in and then begin matching up the pairs and as well code for the few next bases. And for the leading strand, it's just going to continue as long as this DNA needs to be replicated. So it's pretty simple. All you have to have is a primer to begin the process, and then the DNA polymerase just kicks in and begin coding for the rest of the nucleotides. Now, as for the lagging strand on the bottom here, the process is a little bit different. Since the structure of the DNA is an anti-parallel strand with one side going from fifth carbon to the third carbon and one side going from the third carbon to the fifth carbon, the process is a little bit different. You would have the primer installed, of course, here for example, and once the primer is installed by the DNA primase, the DNA polymerase comes in and begin coding for a small segment within the DNA structure. Now, the part that is distinguished from DNA, the lagging strand and the leading strand, is that the DNA polymerase only codes for a small segment, and then after that small segment is coded, another primer is set in again by the DNA primase. So you'd see parts where it's a primer here and parts where it's coded by the DNA polymerase, and back at again with a primer, and then of course the DNA polymerase is going to go in again and code for another small segment. And you can see how it's kind of like repeated between the primer, a DNA polymerase coded section, and another primer and DNA coded section. And for each grouping where you have a primer and a DNA polymerase coded section, we call them Okazaki fragments, discovered by the scientist by the name of Okazaki, of course. And by now, at this stage, both strands are pretty much fully coded, where one strand at the top, of course, is going to have the leading strand, and the strand at the bottom here is going to be the lagging strand. And the distinguished difference here is that the lagging strand has a lot more primers to it. Now, this is when another special enzyme called the exonuclease, which is going to go in and break down all the primer within both the leading and the lagging strand. And basically, it's going to be removing all these primer. Once it's removed, the DNA polymerase then go in and start filling up again. Now, both strands at this point should be fairly identical to one another. There's only one final step in to complete the replication process, and that is for the DNA ligase. And what the DNA ligase is going to do is that it's going to be going over the DNA structure for both the strands, and it's going to begin, of course, sealing up the phosphate backbone in which we have broken down at the very start. Now, you can think of this as pretty much like a stack of paper, in which at the beginning the topoisomerase is pretty much acts as a catalyst. You can think of that as water, for example. And what it does is it makes the paper easier to tear down. And pretty much your hand, you can think of that as the DNA helicase, which tears things in two. And that's pretty much the process of DNA replication.
So welcome back, guys. I hope you guys did not get lost along the way that Thang has just explained the process of DNA replication. So Thang, can you sum it up with a fun example?
Of course. Well, you can see the process of DNA replication as kind of like cloning you see in a sci-fi movie, where the DNA helicase breaking up the DNA and kind of scanning through the DNA is pretty much like the advanced machine and technology scanning a human body or a creature, preparing for the cloning process. And you can think of a DNA polymerase coding for the nucleotides and pretty much forming the two strands of DNA as another complex machinery building the clone using the original creature kind of body as a template. And your end results are going to be two identical DNA strands. And that's pretty much like the two identical clones within the sci-fi movie that you see.
So those clones will then make up the army, which is your body, right?
Yep.
And so before we get on to the next process of protein synthesis, we must first know the three basic types of RNA, or you can think of it as half a strand of DNA, RNA. So basically, Thang, what are the three strands of RNA again?
There's three types of RNA. You have messenger RNA, which can be abbreviated as mRNA. You have transfer RNA, which is tRNA, and you have lastly ribosomal RNA, which is rRNA. As for mRNA, messenger RNA, you can think of that as the instruction paper for the process of making protein. And as for tRNA, you can think of that as pretty much the ingredients, the parts that carries the proteins to the ribosome, so then protein synthesis process can occur. And lastly, you can think of RNA as part of a ribosome that builds the proteins and pretty much contribute to the process of protein synthesis.
Okay, now that makes sense, and let's not waste any time and let's get straight into Thang's lessons.
Alright, now that we have gone over a few kind of the basics, fundamental process, such as the process of the DNA replication, as well as the three roles of the RNA. You have the rRNA, and you have the tRNA, you have the mRNA. We're going to be going to something a little bit more complex, something crucial to the body and something that we all need. And that is going to be involving the protein. So that's right, we're going to be going over the process of protein synthesis. And to break down the process to two smaller kind of major groups, we're going to be having the process of transcription, and we have the process of translation. And altogether, these two are the two major steps for the process of protein synthesis as a whole. Now, focusing on the process of transcription. Now, the transcription begins with the binding of an RNA polymerase, and the RNA polymerase is basically another enzyme similar to a DNA RNA polymerase that pretty much codes for the missing nucleotides. Now, these RNA polymerase are together with one or more general transcription factors. Now, transcription factors, you can think of them as other enzymes. So, other enzymes such as transcription factor 2D, transcription factor 2A, transcription factor 2B. These are all examples of transcription factor, and they are pretty much the enzyme necessary for the initiation process. Remember, the transcription process is divided into three main processes. We have the initiation, we have the elongation, and last but not least, we have the termination process. And pretty much at the beginning here, you will have your DNA strand laid out. And these RNA polymerase and transcription factor is going to locate themselves within a very specific location in the DNA. We call that location the promoter, since within the promoter is a very specific DNA sequence that can be decodable as well as the initial points. You can think of this as pretty much the starting points where you have your enzyme and transcription factor here and there, and you have your RNA polymerase. So this is your starting point. Now, once you pretty much have your everything set up and with all the necessary subunit and unit, and as well as transcription factors and other, of course, enzymes are all laid out, we now move to the process of elongation. And this is when the RNA polymerase begin going through along with the DNA helicase and start cutting the DNA strand into two individual strands. And this process is very similar to those of DNA replication. Now, once the DNA helicase has cut through the DNA strand, you now have basically two individual strands. However, they're connected at both ends, and the connecting point, I don't think I've really talked about that, is what we call a replication fork. So these initial points right here will be called the replication forks, and this kind of bubble that pops up here will be what we call the replication bubble. Now, this is when you have your enzyme, your RNA polymerase, which is going to go through the process here and begin reading the existing DNA codes. And let's say the DNA codes you see so far is A, D, C, C, G, C, A. What it's going to do is kind of read this section here, and it's going to find other bases outside of the nucleus, inside the nucleus. So bases that are floating around, it's going to take that, it's going to read the code here, and pretty much it's going to fill up, and as well match it with a complementary pair. Now, since we pretty much know the complementary pair already, since A matches with T, we're going to have it as U. Since we're dealing with RNA strand, we have a thymine replaced with a uracil. For T, we have it matched up with A, C, G, G, and then C, and then G, and then T. And now pretty much you have this small segment, and what we call that is a pre-mRNA strand. And this process continues on as long as the whole entire gene sequence is completely coded. And what this means is the DNA helicase will then keep cutting, this enzyme then goes on and keep reading, and then this kind of RNA strand keeps getting coded, becoming a longer and longer strand. Now the top strand here is what we call a template strand, and this one down here is the non-template strand, and this non-template strand won't be touched at all during the process of transcription. Now, pretty much at this point, by the end of the transcription process, when the RNA polymerase has fully coded the entire pre-RNA strand, and then there's nothing left for it to do, it goes back to the state where it detaches itself from the DNA, and the DNA goes back to its original form, since during the process of polymerization itself, not only is it cutting along the way, but it's also sealing at its back end here. Therefore, it's cleaning up as it goes. Now, by the time you have your finished product, which is your pre-mRNA segment, which is pretty much the main thing you're trying to get here, you also have your DNA strand go back to its normal position, and last but not least, your RNA polymerase pretty much dissolve itself from the DNA strand and pretty much goes back to the nucleus. And what you want from this process here is your precious RNA strand, but it's not really an mRNA strand yet. It's what we call now a pre-mRNA strand, and there's a reason why. Here you can see a very simplified picture of an RNA, and there are parts to it, such as exon and intron. And these shaded area that I'm kind of drawing right now would be what we call the exons, and they are what codes for the amino acids. So they are the necessary part for the protein synthesis process. However, these other parts that are unshaded are intron. They're basically useless during this process and are eliminated from the RNA strand. Now, once you eliminate that, this kind of finalization process also adds a 5' end cap to the top of the mRNA strand, and it also adds a prime 3 poly A tail, and this will be pretty much the signal for the start and stop for the protein synthesis process. And by this point, with the cap and the tail, and that all your introns here are removed, you basically now have made a fully functional coded mRNA strand. And that's pretty much the process of transcription, guys. Alright, now that the transcription process is pretty much done and you're left with an mRNA, this mRNA pretty much will then leave the nucleus and start entering the cytoplasm and into the ribosome, where you can begin the translation process. And this is the second process to the protein synthesis process as a whole. But before we get into the complex kind of stuff, we need to go over the basics detail. Now, here is pretty much the structure of an RNA. First, you would have an RNA layout, and of course, it's made out of nitrogen and space. For example, you have U, A, C, G, U, C, A, G, and so on until the RNA is pretty much completed. And for each 3 nitrogen space, they are grouped up and called with a special name, and we call them codons. And these codons are then matched up with anticodons, which we'll get into detail later. So for every single 3 bases, you would have what you call a codon, such as this, this, and this. Now, here is when anticodons come in. Now, this is pretty much the basic structure of an mRNA, but what about a tRNA? And within the tRNA, you would pretty much have a structure such as UAC, for example, and this pretty much is similar to a codon with 3 bases. And a transferred RNA will also have an amino acid attached to it. And pretty much the reason why it's called an anticodon is because, for example, you would have a codon with the code of A, U, G, and then these anticodons are going to be matched up complementary to it, since the opposite of A is T, which is U in this case, as we're dealing with RNA, and the opposite of U would be A, and the opposite of G will be, of course, C. And that's pretty much the reason why they're called anticodons, as the codons are pretty much the codes, the anticodons are the opposite of the codes, which are pretty much the complementary kind of sets that goes along with it. And that's pretty much a relationship between these bases. And remember, guys, A always matches up with U, U matches up with A, C matches up with G, and G matches up with, G matches up with C. Since we're dealing with RNA here, this is supposed to be a thymine for DNA, but now it's been replaced with a uracil. Now you guys get the kind of idea between codons and anticodons, and pretty much the tRNA as well as an mRNA. Let's go over into the shapes and what really happens with the ribosomes. Now, first you would have the shape of a ribosome, and it looks similar to what I have drawn here. You have this part down here would be called the small ribosomal subunit, and this top part up here would be called a large ribosomal subunit. And pretty much at this top spot, there's also 3 very special sites. We have the E site. We have these are the small ribosomal subunit, and back to where I was going. We have the E site, we have the P site, and we have the A site. And these are the 3 sites in which the tRNA is going to enter as well as leave later on. And as for the codons, which are located within the mRNA, is going to go through along the middle here. So what pretty much happens is you're going to have, of course, the mRNA with the cap, and of course the tail. And basically, an mRNA would have a certain kind of codes such as, for example, here, U, A, C, A, C, C, and so on. And these anticodons are just going to see what they matches up, and they're gonna come in and they're gonna leave their amino acid as they matches up. So once they both of them match up as this one, let's say if this has U, A, and C, this anticodon will have A, U, and G. As they match up, the codon moves alongside, the new ones go to the next position, and these anticodon pretty much leave. But the only thing they left behind is this amino acid. And once that transfer or tRNA leaves, pretty much a new tRNA comes in and then start doing the same process, and then this one comes in next and next. So another pretty much example of this would be here you would have your large ribosomal subunit, and let's say your codon is a U, A, C. Then by this time another tRNA is going to come in with anticodon, which is A, U, G, and once it matches up, it leaves the amino acid behind, and then it enters or acid through the E site. And these kind of amino acids that are left behind will then stack up with the next one, and eventually you would have a similar structure to something as, for example, something such as a structure that then just keep building up with amino acid after amino acid. And once this one, of course, leaves, this amino acid will then put on into the upcoming next one with the next codons along the line. And of course, there are signals which indicate the start and, of course, the stopping of this whole entire translation process. You have codons such as A, U, G, which pretty much starts or begin the translation process. And you have other codons such as UAA or UAG or UGA, and these codons indicate the stop of the process or pretty much the end of the mRNA segment. And pretty much once the ribosome sees that AUG, it knows that the process is beginning, or when it sees one of this, it knows that the process is ending. And with this, like I've said, as I was going, as the amino acid chain is building up, eventually, of course, it's gonna end up with a final tail where it's gonna meet up with one of the ending codons, and that means it's time to stop. And by this point, there's no more tRNA coming in, there's no more mRNA coming in, and these codon, this amino acid will basically pretty much folds together and form a protein, or they can went through more further modifications. But that's pretty much a simple translation. From what you have to begin with is a strand of mRNA, you have a bunch of tRNA with amino acids, and of course you have your shape similar to ribosome. And pretty much this too enters this processor here, and pretty much what you get at the end is a very complex kind of proteins, and these proteins are made out of chains and chains and chains of amino acid. And that's pretty much the step of translation.
Back at it again. I hope you guys did not